Hello everyone. So on today's video, we'll be taking some high-yield examination question in biochemistry, and we'll be focusing on carbohydrates for today. Let's begin. For our first question, so for each question, I'll give you about 10 seconds to think of the answer before we move on to the next one. So your time starts now. Which of the following monosaccharides is a ketohexose? This is a simple one, and the answer is fructose. As you know, fructose is a ketose, and ketose sugar contain the ketone group. But for fructose in particular, it has the ketone group as the C2. The C means carbon 2 at the carbon 2. Our next question. What is the reducing sugar that is formed when lactose is completely hydrolyzed? Hydrolyzed means when you break down a bigger compound or molecule into smaller constituents using water. So what is the answer? What will you get when you hydrolyze lactose? The answer is A, glucose and galactose. So you get glucose and galactose, and the glucose and galactose, they are linked by beta 1,4 glycosidic bond. Now, glycosidic bond is kind of bond that is found that holds different sugars together. Now, to get a disaccharide, you have to have two monosaccharides. Now, these two monosaccharides are being held together by a glycosidic bond. In my next video or future videos, I will try to break down for you what the beta and the alpha represent, but not in this video. For our next question, which enzyme catalyzes the first committed step in glycolysis? You know, this the stages in glycolysis pathway. Which enzyme will catalyze the first committed step? And the answer is B, phosphofructokinase. So this step is a rate limiting step. What does it mean? It means that if this step is affected, the other steps that follow cannot proceed. Our next question. Glycogen phosphorylase removes glucose residues from glycogen by what? A, hydrolysis producing free glucose. B, phosphorylases producing glucose 1-phosphate. C, oxygenation to glucose 6-phosphate. D, hydrolysis producing glucose 6-phosphate. C, acetylation and removal. So which of these answers will be the correct one to this question? Now answer is B, phospholysis, which will produce glucose 1-phosphate. Now for this reaction, it uses inorganic phosphate instead of water. For the in our first question I mentioned hydrolysis, but for here they are using phospholysis. So you are breaking down the molecule with phosphate, not water. If it was water, it would be hydrolysis. For our next question, which of the following best describes the Krebs cycle? A, glycogen synthesis from glucose, glucose synthesis for alkaline in muscle. C, lactate produced in muscle is converted to glucose in liver. D, direct conversion of fructose to glucose. E, glycogenesis in liver during fasting. So which of the following best describes the entirety of the Krebs cycle? So what is your answer? What do you feel the answer is? There are some pretty good options there, but there are some pretty bad options which you immediately eliminate from your mind. And the answer is C. Lactate production in muscle is converted to glucose in the liver. So the muscle will produce lactate during anaerobic exercise, because during exercises, especially aggressive exercises, body doesn't have enough time to take in enough oxygen, and even the little oxygen that is taken in is consumed immediately. So it's basically an anaerobic process during that time. For our next question, lactose is primarily metabolized through which process? Option A, direct glycolysis. B, conversion to glucose phosphate via lactokinase and galactose 1 phosphate uridotransferase. C, oxidation to CO2 and H2O. D, conversion to fructose 1,6 biphosphate. E, pentose phosphate pathway only. This is your answer. For our answer here is the conversion of glucose phosphate via galactokinase and galactose 1 phosphate uridotransferase. So this B is the answer, and it's kind of obvious if you know the pathway well. Moving on to the next question. The primary storage form of carbohydrates in animal. This should be a 2-second, 1-second question. Very simple. The primary storage form of carbohydrate in animal is what? The answer is glycogen. Answer is glycogen. Why? So in animals, the main storage is glycogen. So, but this question can be twisted, and it can be main form of storage of glucose in plants is what? The answer will be starch. Yeah. For animals, it's glycogen. So, and a sub question: which enzyme breaks down glycogen? Glycogen. So think of the answer. You know, when there is excess sugar, as glucose in the body uses insulin. Now insulin forms glycogen. It helps to convert the excess glucose to nitrogen. So excess glucose to glycogen in the liver, so that the blood glucose level doesn't go too high. That's what prevents diabetes. But in people who lack insulin or are resistant to insulin, they tend to develop diabetes. So the next question. Question 8. Which enzyme removes the outer glucose residues in the debranching of glycogen? Which enzyme removes the outer glucose residues in the branching of glycogen? A, glucose synthase. B, glycogen synthase. B, glycogen phosphorylase. C, the branching enzyme. D, alpha 1,6 glucosidase. E, both C and D. So which of these will remove the outer glucose residue in the branching of glycogen? And the answer is E. Both C and D do the job. So both the debranching enzyme and alpha 1,6 glucosidase will remove the outer glucose on the molecule. So that's when they want to debranch a structure from it. For our next question, question 9. The rate limiting enzyme in gluconeogenesis is what? The rate limiting enzyme in gluconeogenesis is what? Just the same way before I asked you what is the committed step. There are kind of synonyms to each other, so they mean the same thing. But for that was glycolysis. This is gluconeogenesis, so do not confuse it. Gluconeogenesis is a different process. So for here, you should have read the options by now. What do you believe is the answer is? The answer here is C. So gluconeogenesis is a process. It occurs in the liver whereby glucose is formed. Normally we get glucose from the food we eat, but now let's say in conditions such as starvation, the body might have to form its own glucose in order to maintain cellular function. Now for question 10, which is our last question but not the least, in the pentose phosphate pathway, which enzyme catalyzes the rate limiting step? In the pentose phosphate pathway, which enzyme catalyzes the rate limiting step? A. Transketolase. B. Glucose phosphate dehydrogenase. C. 4-phosphogluconate dehydrogenase. D. Transaldolase. E. Ribulose 5-phosphate isomerase. So what is the answer to this last question? I kept the toughest for last. It's simple, but kept the toughest for last. And the answer is B. Glucose phosphate dehydrogenase. Now, this particular enzyme is very important. It causes, when there's a deficiency of it, it causes hemolytic anemia. Why? Because this enzyme helps to maintain and to reduce the level of oxygenation of red blood cells. When there's excess oxygenation, the red blood cell tends to lyse and break down due to excess oxidative stress. So that will be all for this video. Kindly subscribe. Thanks for your support. See you soon in my next video.